In this lecture, I'm talking about DNA. We're going to talk about the basic structure of DNA, part of which is a review from the nucleotide lecture. And then we're going to talk about how DNA replicates, how DNA makes a copy of itself so that we can make two new cells during cell division. So let's start by just looking in a little more detail at the structure of DNA. Remember that DNA is a polynucleotide, so it is a string of nucleotides. And here's the basic nucleotide structure. Remember that it's a pentose sugar, and in this case we're talking about DNA, so our pentose sugar is deoxyribose. There's a base and a phosphate group. Then these string together to make a polynucleotide. In DNA we have two polynucleotide strings that run anti-parallel to each other. So what I mean by that is when we string these all together, one runs this direction and the other one runs essentially this direction. You can see that one is upside down relative to the other. So two anti-parallel strands. You should also notice that here, this base can be one of 4 different bases for DNA. There are 4 different bases that can be in that location. In other words, there are 4 different nucleotides that string together to make the DNA strand. So 4 different bases. Adenine, guanine, cytosine, and thymine. And we usually just represent those by their first letter. A, G, C, and T. And that's what you see. This is the more cartoony version. A, T, G, and C. You notice that the 2 strands are connected to each other at the base that sticks out from this nucleotide. And you should also notice that there's a pattern here. If you look at who's connected to whom, you'll see it's always the same. The same 2 bases always connect with each other. So here's the rule. A always bonds with T, and C always bonds with G. So wherever we have a T, there's an A on the other side. Wherever there's a G, there's a C on the other side, and so on and so forth. You should also notice that they are connected together by these 3 dots. Do you remember what type of chemical bond we represent with the 3 dots? It was a hydrogen bond. So the bases are connected on the 2 anti-parallel strands by these hydrogen bonds. What do you remember about hydrogen bonds? Are they really strong and hard to break, or are they relatively weak and easy to break? They're very easy to break. So this is a good setup. We need those bonds to be easy to break because we need to separate those 2 strands so we can get in there with an enzyme and read that sequence to copy it. And we're going to do that by breaking those hydrogen bonds. One other thing you should note is if you look at that carbon, you notice there's an oxygen at the top. And really, I've represented this very basically. Really, there's a carbon that connects to this phosphate group. And there's a carbon here as well. So if we count the carbons that make up this pentose sugar, we know there should be 5 carbons. That's why it's called pentose, meaning 5. And if we counted those carbons, starting at the oxygen and going clockwise, it would be 1, 2, 3, 4, 5. 5 carbons. So looking on the screen, 1, 2, 3, 4, And then the 5th one is actually outside the ring, and that's what connects to the phosphate group when we draw it with a little more detail. That is significant because these two anti-parallel strands have a specific direction, and we refer to that direction based on which carbon is at the top of that strand. So in this case, if we look at the strand going this way, which carbon is at the top? 1, 2, 3, 4, 5. It's the 5th carbon. So we call that the 5' end of the DNA strand. The 5' end of the DNA strand has that 5th carbon at the top. This is the 3' end because it has the 3rd carbon right there. 1, 2, 3. And then this one runs the opposite direction. So when these nucleotides string together, they're going to have a specific direction. One is going to run 5' to 3', and the other is the opposite. This is significant because we're going to copy this DNA continuously on one side because it can only be replicated continuously in one direction. On the other side, it's going to be discontinuous. And really, we should draw these arrows such that they show these going opposite of each other. So if we show the carbons facing up, this would be more accurate. Running opposite of each other. Now let's look at the details of how this DNA is going to make a copy of itself. But before we do that, I want to remind you of when we make a copy of DNA and why we need to make a copy of DNA. And I want to make an important distinction between this and making messenger RNA. So there are going to be 2 times that we need to get in and read this strand of DNA and make a copy of it. One is going to be during DNA replication, and the other is going to be when we need to make a protein. Remember, this DNA represents the recipe for how to make a protein. DNA replication. It happens only once during the life of a cell. This is an important distinction because we're going to be making messenger RNA continuously. We're constantly needing to make protein. So we get in and read that DNA recipe on a regular basis to make messenger RNA. But we're only going to replicate this DNA once during the life of a cell. When is that once? Well, let's remember the cell cycle. G1, S, G2, and M. When does DNA replication happen? It happens during the S phase of the cell cycle. That's when DNA replication happens. So it only happens once during the life of a cell. Remember, what is happening is in G1, every chromosome has just one chromatid. And what we're doing is we're copying that chromosome in its entirety and we're attaching a copy. Why? It's so that these can get pulled into 2 separate cells during cell division. So that's what DNA replication is. It's making this copy of every single one of our chromosomes. So it happens only once, and we copy the DNA in its entirety. All the chromosomes. Because we want to make 2 genetically identical cells. So we need to copy all the chromosomes in their entirety. How do we do this? Well, let's imagine a strand of DNA. I'm going to give a very cartoony version this time for our strand of DNA. I'm going to draw just a line to represent the pentose sugar and phosphate group all the way down the side. So this would be the side rail of the ladder. And we have our 5' end and our 3' end. And then I'm going to have the bases stick out as the rungs of the ladder. And I'm going to make an imaginary made-up base sequence here. So we have 4 possible bases. So if one side looks like this, what would the complementary side look like? Remember complementary base pairing. A and T always go together and C and G always go together. So on this side, wherever there's a C, it's going to be bonded to a G. G would be C, A would be T, and T would be A, and so on and so forth. I'm not going to draw all the hydrogen bonds connecting these together because we're just going to need to break those bonds in a minute. So this would be 3' and 5' end. Again, that has to do with the numbering of the carbons on that pentose sugar. Now it's time to copy this strand of DNA. We need to copy both sides because we're making an exact copy of that whole chromosome. So we need to copy both sides in their entirety. One option would be to come in and make an exact copy all the way down. I'm not going to write the whole sequence, I'm just going to do part of it so you can see. Okay, and then remember that we connect these two sister chromatids. They're sister chromatids because they're genetically identical, and we would connect the two with these cohesin proteins in the middle. And then during mitosis, anaphase of mitosis, these get pulled apart and they move into 2 separate cells. This would go into one cell and this would go into the other. In that case, this would be our old DNA and this would be our new DNA. So one of those cells would be getting all old DNA. The other cell would be getting all new DNA. Not really good either way because this one could have suffered mutations just by being exposed to things like UV radiation, environmental toxins. And this one, being all new, we could have made some mistakes in copying this DNA. Mistakes are rare, but they do occur. Another option would be what really happens. Okay, so this is not what happens. We don't make one strand completely new and one strand completely old. Instead, what happens is these two split apart and we're going to copy each side so that our new DNA is really half old, half new. It's called semiconservative replication. Half old, half new DNA on each resulting strand. And here's what that looks like. Okay, but before we do that, just a quick shout out to Rosalind Franklin. She is who discovered the structure of DNA through X-ray diffraction studies. She ended up getting cancer probably from her research, and she died before Watson and Crick were awarded the Nobel Prize for discovering the structure of DNA. Watson and Crick really didn't discover anything. They used other scientists' research to put together a model of DNA. You are not eligible for the Nobel Prize if you are not living, so she never received the Nobel Prize, but she should have. She is who actually discovered the three-dimensional structure of DNA, and this is her famous photograph. So this is what happens during DNA replication. I just said And we don't end up with all old on one side and all new on the other, so that one cell is getting old DNA and one's getting new. It's called semiconservative replication, and here's how it goes. Here's what we call the parent molecule. Okay, so this would be our original DNA. And it's going to split apart. We're going to break these hydrogen bonds. So separation of the two strands. And then we're going to copy each side. So let's do that on the board with this original sequence that I gave. I know it seems tedious, but it's very, very important to understand how this happens. So let's split this apart. I'm only going to do part of the strand. Here's the other half. So remember complementary base pairing, G, C, T, A, T, C, G. So we've split this apart. And so on and so forth. Now we're going to copy each side. So anywhere there's a C, our enzyme is going to read this, and it's going to attach a G. Anywhere there's a G, a C, A, T, T, A. So you're starting to see this new strand we're making looks just like this side. A, C. Same thing's going to happen on this side. It's just going to happen the other direction. Again, this is going to look like this side. Our new hydrogen bonds. And now what we end up with when we connect these two is we end up with half old, half new on each strand. And that's called semiconservative replication. This is a good setup because now we have one side that has actually already gone through cell cycle checkpoints before it divided. It went through especially that important G1 checkpoint that checks for DNA mutations. So the old DNA has already been through a checkpoint. Now we've copied it, hopefully correctly, and we have half new DNA. That will go through a checkpoint when it goes through G1 after cell division. So semiconservative replication. How do we do this? How do we make this copy of the DNA? Well, that gets more complicated. I'm going to give you a very basic version of this story. You can plug in details later if you take a higher level biology class. This is going to be a very basic version of the story. What do we need to replicate DNA? We need something to copy, the parent molecule, and we need a suite of enzymes. There are going to be some key enzymes in this story. The first thing we need to do is separate the two strands from each other. Break those hydrogen bonds and separate the two strands. And that is the job of an enzyme called helicase. Helicase unzips the two strands of DNA. Then an important enzyme is going to come in and read that strand and bring in nucleotides to connect in a complementary fashion. Before that can happen, though, that enzyme needs to know where to start reading, and that's the job of an important enzyme called primase. Primase is going to lay down something called an RNA primer. It's a short sequence of nucleotides that are actually RNA instead of DNA, complementary to the DNA, but it's an RNA primer. This is going to tell the next enzyme, which is called DNA polymerase, where to start replication. Okay, and then DNA polymerase, again, we know these are all enzymes because they end in ASE, and DNA polymerase is what's going to form the DNA polymer. Love enzyme names. Again, they tell you what they do. DNA polymerase is going to make the DNA polymer. This reads the DNA on one side and attaches complementary bases, complementary nucleotides. There's going to be another important enzyme, because what's going to happen is on one side we're going to be able to copy continuously. On the other side we can't copy continuously. It has to be looped out and copied in fragments called Okazaki fragments. And that's the job of the last enzyme called ligase. Ligase is going to be the enzyme that stitches together the Okazaki fragments. I'm going to define Okazaki fragments when we talk about this discontinuous replication on one strand. We'll talk in a little more detail about that. So these are the key players in the story. So let's go through the story on a very basic level. So we start with what we call the parent molecule. We start with this original strand of DNA. That original strand of DNA is going to feed through the helicase enzyme, which is going to unzip the two strands, break the hydrogen bonds, and they're going to start spooling out in two separate directions. One of these strands is going to be called the leading strand, and one is going to be called the lagging strand. Okay, there are also some important protein complexes that attach, that keep the two strands apart. And then we're going to start copying the DNA. So let's look at the next slide. Oops. Here's what's important. The leading strand, what we call the leading strand, is going to be the strand that's copied continuously. Why is this copied continuously? Well, that's because DNA polymerase can only read and copy continuously in one direction. And you don't need to memorize which direction this is. At this point, you just need to know that the leading strand is the one that's copied continuously. So the leading strand is copied continuously. If you look at the figure on the board, you can see on the screen, you can see this leading strand is the continuous one. That's because this DNA polymerase can only read 3' to 5' continuously. You don't need to memorize that at this point. It's not that important. At this level of detail, it's not the most important thing. What's important to note is that DNA polymerase is the enzyme that's going to read this strand, and it's going to attach complementary nucleotides. It's going to do it continuously on this strand. This strand is discontinuous. It's called the lagging strand. So lagging strand is not copied continuously. Instead, what happens is it's looped out and it's copied in fragments. So copied in fragments, and those are what's called the Okazaki fragments. So that's what you see on this side. looped out and it's copied in fragments because we have to go backwards on this strand. DNA ligase is the enzyme that's going to stitch together those Okazaki fragments. There's a little more detail on this next slide. Again, this is a very basic level. If you take a higher level biology course, you can plug in more details later. I just want you to know the basic story. So this is the leading strand, the lagging strand. This helicase is what unzipped these two strands. Here are the proteins that are keeping them separate. The DNA polymerase starts reading. How does it know where to start reading? It's because an RNA primer was laid down by this enzyme called primase. Primase is going to lay down this primer. It says to DNA polymerase, this is where you start reading. Please don't pay any attention at this point to DNA polymerase 3 versus 1. They do serve two different roles, which you don't need to know at this point in time. This is the lagging strand. So we have fragments that are going to be stitched together by DNA ligase. I know this isn't very much detail, but here are the things that are important to know about DNA replication at this level. It happens once during the life of a cell, at the S phase of the cell cycle. What is the purpose of it? The purpose is to copy every chromosome in its entirety so we can attach an identical copy. So here's chromosome number 1. We're going to make an exact copy and attach it so that we can pull this into two separate cells during cell division. It only happens once, and we copy the DNA in its entirety. The goal is to have two genetically identical cells. So it's important to understand one strand is the leading strand, copied continuously, one is the lagging strand. Why? Because DNA polymerase can only read the DNA in one direction. The ligase is what stitches together the Okazaki fragments. We'll add on the lagging strand. And then remember, these were the other important enzymes: helicase, primase, DNA polymerase. Okay, in the next lecture, we're going to talk about the other time we come in and read the DNA template, and that is to make messenger RNA so we can make protein.